Hey everyone, thanks for joining us today for episode 691 of People Behind the Science. I'm your host, Dr. Marie McNeely, and I'm excited to introduce you to today's guest, Dr. David Kaplan. Listeners, if you've been enjoying our podcast, it would be fantastic if you could share our show with a friend or subscribe, rate, and review us on your favorite podcast platform. And today, David has some outstanding stories of his own to share with us after a short sponsor message.
Looking to begin a custom antibody project? Lampire Biological Laboratories has over 3 decades of experience and is housed 100% in Pennsylvania. Call us today at 215-795-2838 to speak to your project manager, or you can visit lampire.com.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Listeners, today I am excited to be speaking with our guest scientist, Dr. David Kaplan. So, David, welcome to our show today. How are you?
I'm doing very well. Thanks for having me today.
Well, thank you so much for joining us. We're looking forward to learning more about you and the wonderful work that you do, David. But let me first start by telling our listeners a little bit more about your current position and your background. So listeners, David is the Stern Family Endowed Professor of Engineering at Tufts University, a distinguished university professor and a professor and chair of the Department of Biomedical Engineering at Tufts University. He received his bachelor's degree from the State University of New York in biology and his PhD in biochemistry from Syracuse University and SUNY Syracuse. He worked as a research scientist at the Natick Research and Development Center for several years before joining the faculty there at Tufts University in 1996. Now, David has received multiple awards for his research and teaching, including the Clemson Award for Literature Contributions from the Society for Biomaterials, the Massachusetts Columbus Quincentennial Award, and the Henry and Madeline Fischer Faculty Award from Tufts University. In addition, he is an elected fellow of the American Institute for Medical and Biological Engineering, the National Academy of Engineering, and the International Academy of Medical and Biological Engineering. And today, David, we're excited to get to know you as a scientist, but also as a person. So can you start by telling us what you like to do when you're not doing science?
So when I'm not doing science, which is not very often, I enjoy traveling, seeing the world, obviously meeting people. But probably my favorite hobby is observations of nature. And my favorite topic there is studying trees. And I mean that from just a naturalist perspective and a scientific perspective as well. Just trees to me are one of the amazing living creatures on the planet, and understanding their role and how in tune they are to their environment, their surroundings, what they contribute, linking the earth to the air as an interface. I just find them remarkable in their structures, in their functions, and in their inspiration.
Phenomenal. So are you the sort of person where someone can bring you a leaf and you can tell you what kind of tree it came from?
I did study dendrology when I was in school, so I could tell you some of that. It's been a while, but to me, it's more looking at perhaps the distribution network in the leaves and seeing how well things are integrated into a system to have a functional leaf that collects sunlight, produces sugar, and supports the tree. I mean, those are the things that fascinate me.
I love it. Do you have a favorite kind of tree?
I do, of course. The giant sequoias in California are my religion, to the ridiculous point now where I've planted what are called dawn redwoods in my backyard here in Massachusetts, because they grow in this area. They don't grow to the size of giant sequoias, but they do grow in our climate, and they give me a sense of comfort being out in the backyard.
I love it. Well, it's great to get a window into what you're passionate about outside of your specific research, but I'd love to talk about your scientific side as well. So, David, how do you describe your work to people who aren't in your specific field or maybe are not in science at all?
I guess I would say kind of a segue from what I just described about my interest in trees. What I've always tried to do is look at nature for sources of inspiration and then borrow from that and use our scientific and engineering disciplines to try and make something new, right? So whether it's looking at polymers from nature, we study things like silks from spiders and silkworms. And so observing an orb web and its function is just a remarkable feat of engineering. But then we take it to the lab and try and understand the molecular biology, the biochemistry, the material science from that. And that applies as well to tissue engineering and regenerative medicine, where we look at how tissues are functional in vivo and what can we do then to create those kinds of systems in the laboratory to really study and understand them. So really it all starts from nature, just appreciating the wonders of what works out there and then trying to emulate that in some way in the laboratory.
Definitely. And you have a really wide range of different kinds of projects within your research portfolio, which I find fascinating, all the way from, as you mentioned, regenerative medicine to kind of some of the more material science side of things, all the way to cellular agriculture. So how do you balance or I guess maintain all of these different kinds of projects?
I kind of look at it as an evolution, and really the core of all of those fields is the biomaterials side. That's really my foundation, which really starts with understanding polymers that exist in nature, from the silks I mentioned already to cellulose and collagens and keratins and many others. And first trying to understand how they're made and then to utilize tools to either genetically modify or control them in different ways to make new structures and new functions from those structures. And once you start to do that, you then have a sort of a biomaterial template that you can use to house cells. And those cells then become the tissue engineering side of it, right? So the cells generally need a substrate to grow on because the cells are anchorage dependent for the most part. So you need to create an environment for those cells that will support their expansion and differentiation into whatever tissue or whatever disease model you're trying to understand. For us, it could be right now the human gut, it could be the human brain or human skin as current examples. So that all starts with really designing the right biomaterial substrates. Could be 1D fiber-like, 2D film-like, 3D sponge-like, and then creating that habitat, if you will, for the cells to grow and expand and create a tissue. And that applies also to the more recent efforts in food. It's the same idea. It's still tissue engineering on biomaterial substrates, but now the cells are animal-derived for foods as opposed to human-derived for regenerative medicine and those kinds of medical goals. So I think there's actually a common theme there, and the group has evolved. The research activities have evolved from strict biomaterials many decades ago to human tissues over the last 20 plus years, and now the last 5-10 years into tissue engineered foods as well for cell ag.
Well, David, this is so cool. And I look forward to diving into the detail of some of these projects as we go through our conversation. And I think motivation is really key for success in science and perhaps to help motivate and inspire our listeners out there. David, do you have a favorite quote or a saying or maybe something that just really motivates and inspires you in the work that you do?
Well, I have a number of standard lines I bestow on all my students, whether they want it or not, right? And the one thing I'll always say is don't do anything boring. And what I mean by that is if we're going to do research, if the student's going to really invest their time and energy, their heart and soul, I want them to do something that's going to be pioneering and really push the boundaries in a way that will make a meaningful difference. That's something I try and live by. We want to really continue to learn more and push forward because there's so much we don't know and so many opportunities to learn and then apply that learning to make the world a better place through the materials, through the foods, through the tissues that we and others try and grow.
I love this. I think there is so much exciting science out there yet to be done. So why settle for something boring, right? Well, that is wonderful. And as a scientist, I know there are a lot of people, perhaps like yourself in the case of your students, who motivate and inspire you along your journey. So when you think back on your own career, your own life, David, are there particular people who really rise to the top of your mind as mentors or role models or people that just really had a big impact on you?
For sure. I think we all learn from people along the way, and inspiration is kind of a hard thing to define. But I would say the most impactful person, inspirational in the impact of my career path into research and as a faculty, was really a professor in my undergraduate days. Not so much for the research we did. We were studying salamanders back then at University at Albany. But once we had all the data, I remember him taking the time to sit down, and together we kind of crafted the paper, the publication from the work. And I think it wasn't even the fact that we did that. It was the fact that he took the time to walk me through the process, to mentor me in the process, and to show me how you could take a collection of data that I loved collecting, but didn't see necessarily where that was going to have impact, and turn it into eventually a peer-reviewed publication. So I would suggest it's more the time and attention a mentor, at least in my cases, gave to me that really was impactful. So we try and from that process, I've always tried to make my lab open to undergraduate students to at least give them the exposure to research to see if they like it or not.
Definitely. I think understanding the process of research is really key because it is sort of an abstract thing. You have this idea of like, oh, you're in the lab collecting data, but there's so much more to being a scientist than just the data collection.
For sure.
Now, you were a biology major at that point already, studying salamanders in college. But David, could you rewind back even further and maybe give us some insight into what first got you interested in science and on this path?
If I think way back, there's maybe a couple things that always stood out with me. One is I was always someone who liked to just go hiking out in nature to observe. And whether it was the plants, whether it was the animals, didn't matter. Getting outdoors and experiencing nature was always something I loved. And we always lived pretty close to the water. So that was also something that I felt was really meaningful to me as well. So early days, that's certainly something that had a big impact. And then I also always talk about, I remember back in high school, I think it was second year of high school, I had a teacher who was showing us laboratory experiments. And I remember I felt like I was in a magic show because he took a solution of a certain color and used an enzyme to show the solution change color. Mm-hmm. And today it doesn't seem like a lot of meaning, but back then as a young child, the ability to show you could take a color, reverse it with an enzyme, a protein, and turn it back again was truly transformational to me at that point. So I think I got the bug then as well, that there really were some pretty cool things you could do by starting to borrow parts from nature and use them in prescribed ways to, in this case, make a color change, but in other cases, obviously, to do a lot of other things as well. So I would say those are maybe some of the earlier experiences that either reflected on my interest in these areas or helped shape my interest.
Absolutely. I think some of these early school and lab experiences really leave a lasting impact. These are the memories that you carry with you for a lifetime. And I think that's something that's really important to emphasize to high school teachers, to even college teachers teaching those intro classes. One little experiment, one little statement that you make can be so transformative for students. So I guess what then took you to that point of sort of getting interested and curious about science? What are some of these key moments that really helped you then, David, get to where you are today?
So the journey's been long, but wonderful. Every sort of career step has been important. So as I briefly mentioned, as an undergraduate student at Albany, I was able to experience lab work for the first time. And I think that's really where my passion for doing research came out, because I don't want to generalize, but many of us that end up in a lab environment tend to be a bit introverted, I would say. And I was certainly that way in my younger days. So the lab environment is very comfortable, right, for someone like that. You can do your work. You have a limited number of lab mates around, and you can sort of screen out the rest of the world and focus on things you enjoy. And so that was very meaningful, I think, for me at that point. And when I went to Albany, I wasn't sure. I was either going to be an architect or something else. And I decided on the something else, which was research and science, which worked out great. And then from there, I went on to Syracuse. And there, I really was fortunate. I was in a laboratory that, again, I didn't put two and two together back then, but it was a lab that allowed me to study for my PhD, the synthesis of lignin. And if for the audience that knows anything about lignin, that's one of the key components in trees and plants, right? It's sort of the glue that holds a lot of the components, the hemicelluloses, the celluloses together. So that's where my love of trees, I think, spilled over into my love of science. So I studied the enzymes and the mechanisms by which lignin was synthesized in plants. So that was really where I started moving from what I would call the macro scale research into the molecular scale research. So my career has really been an evolution in that direction through that stage, at least. And then after that, I ended up working at the Army Natick Labs to set up their biotechnology program for a number of years after my PhD was done. And I focused mostly on learning about polysaccharides from a lot of different organisms, fungi, algae, etc., and trying to begin to use what I would call more material science and engineering to manipulate these polysaccharides more for their material properties. And back then I was interested more in sort of things like water transport, mechanical properties, all keeping in mind things like sustainability, edibility, degradability as part of this. So again, the theme always has been materials from nature, understanding them and trying to then exploit them in new ways to make new materials, new structures, and so on. And then at Tufts, it's really grown exponentially into many different areas. We've talked about the proteins already a little bit, so we've expanded our studies here in silks in many ways, and this is where we get into genetics and bioengineering new proteins from genetic principles. We do modeling with colleagues on these as well, and then into the tissues and the foods that I mentioned before. So maybe that was a bit too long and windy, but those are some of the steps I can quickly recall as we built on my early interest in nature and enzymes from high school and before.
Absolutely. And I think this experience that you had working at the Army Natick Labs is really interesting and something that people may not know much about. Can you talk a little bit about what that was like and then maybe why you decided to make that transition into academia?
So after school, my PI back at Syracuse knew one of the PIs at the Natick Labs. And if you're not aware, Natick Labs is just outside of Boston. And I had my older sister, Susan, lived in Boston. So I used to visit her. And I was really intrigued by Boston. I love visiting. I thought it was a pretty cool city as someone who grew up outside of New York. So I was kind of committed to finding my way to Boston. And it turned out my PI made an introduction for me to the guy at Natick. And he was looking for somebody to create a biotechnology research program, which was perfect for me. So took the job, wasn't interested in academics at that point because, again, still being someone a bit introverted, not that comfortable, even being a TA, teaching assistant, I thought a lab environment and research was just right for me. So off I went and I built the program there, I hired a whole staff, and we explored a lot of different polymers, again, towards material properties. Some of these were going to be for food, some for materials for clothing. These are all areas Natick works on and still does today. And a lot of things were fortuitous at the time. You have to be open to things in life that just happened by serendipity. One is I was fortunate to meet my wife at the time. She was an intern there, a student intern. We ended up getting married, and that was a big step in my life, obviously. The other is I always would have summer faculty in who spent their summers at our labs at Natick, and then they would go back to their academic jobs during the school year. And as I got to know them, they would then ask me to come fill in, let's say, for faculty on sabbaticals to teach their classes and so on. And I was always someone, like I said, who never felt that comfortable doing it. But after many years away from it, I started teaching at Framingham, now called Framingham University, and taught everything from nursing classes to recombinant DNA classes, and found out after a couple of years away, I totally loved teaching again. It was just something I really found enjoyable. And so I started doing quite a bit of it every semester, and it just got very difficult logistically to run the lab at Natick, run back and forth to teach, and not be there for the students on a more consistent basis. So that's when I decided it was time to move back to academics. I felt both more comfortable with my own skill sets. I think I'm still always introverted, but less introverted, let's put it that way. And I now had a family and a little more stable lifestyle, so I felt time was right. And my spouse, her only request is we couldn't move from where we were. So I had to look locally in Boston, which was fine. There's a lot of schools in Boston. And just fortuitously, there was a position open at Tufts. I applied for it. It was in chemical engineering when I started, and also to take over a biotechnology core program here that they had started. Sounded perfect for me. And so got the job and been here ever since, about almost 30 years now. And started in chemical engineering. And then about 22 years ago, the faculty decided to form biomedical engineering based on the requests from students and their families. And a group of the faculty, including myself, got together and formed biomedical engineering. And that's, again, where I've been for the last 21 years now.
Phenomenal. And David, I really like one of the points you brought up in this story of just embracing the opportunities as they arise. And I think that's something a lot of students stress about, feeling like they have to have everything planned out in advance. But sometimes you just have to kind of go with the flow and see where life takes you. And I think that's a completely legitimate career strategy in science, because these opportunities will arise and you're not going to know about them years in advance.
I think it's more true than ever today. I mean, when I made the transition from being in a government lab, the Natick labs, when I started my career to moving back to academics, it was very unusual to do that. I was fortunate that the opportunity existed at Tufts and they were open to it, but it was not very common to do that. I think today that's much more commonplace, and to some extent it's almost embraced. And I think the change is good because I think ultimately the students benefit from having faculty with broader experiences that come from different places and can offer different perspectives. And I think that's more true than ever, more needed than ever. And I think that's something we should all embrace. And back to my advice I give my students, be ready for these, I call it the zigzags of perturbations in your career steps, because you look at me, I went from academics to government back to academics. I think those are things that students certainly need to be more open to today. There's very little permanency in industry anymore, whether it's a startup or even a big company. And academic life is very different, but very rewarding, but it has to be right for you. So these are things to explore and decide what really fits. The other personal point I'll reemphasize, although I've already said it, is I think we all hopefully mature, grow, get more comfortable on our own skin with experiences, with maturity, things like that. And so what you think you're going to be doing as a student today may evolve, and it could evolve quickly or it could take a long time, but you may find yourself ready for a change when you least expected it. And my advice to anybody is keep as many doors open as you can. And when you're ready, you'll know the right door to go through. And as a PI, my job is to make sure I provide those doors to my students, but ultimately it's their decision as to which one they're going to use. And that may change over time, and that's a good thing. So I think there's a lot to be said for versatility and adaptability and being open to change today.
Absolutely. I could not agree more. And it sounds like you found the perfect fit for yourself there at Tufts. I know you've got some amazing projects going on in the lab. So, David, is there one in particular that you're perhaps the most excited about at the moment that you'd like to tell us about?
Oh boy, I can't pick just one, Marie, but I could just highlight two maybe to be a little more practical. Mm-hmm. One I'll be very brief on, and that's just our more recent work on trying to develop fundamental tools to grow foods of the future, as we call it. So this is the field of cellular agriculture. And I love it because every step of the way we have to rethink what we've done before, because you have to think about cost. You have to think about animal-free ingredients, and you have to think about non-human cells and components to make the process go. So there's a treasure trove of science to be done, all of which has or will have an amazing impact on sustainability, animal welfare, human nutrition, and just feeding the anticipated 10 billion people on this planet over the next 20 plus years. So we have a lot of work to do. And it's coming fast and furious, but it's really an exciting time. And for the students out there, I can tell you the students in my lab involved in this area, field of cellular agriculture, I have never seen, all my students are smart. That's not the issue, but I've never seen such passion about what they're doing in terms of making a difference. And I think this is really incredibly satisfying as a PI to see students at all levels, undergrad, high school even, and then on to graduate school and postdocs contributing to this work in our laboratory. So that's one area I would highlight for sure, and I'm happy to get into details if there's other questions about it. The other that I think is really, really important is our work in the human brain and our tissue-engineered human brain. And I'll just highlight a couple of things that we've reported over the last 2 to 3 years that I think are very meaningful, particularly in the time of COVID. So I think everyone can relate to these issues. So I'll tell you a short story. First, we developed the human brain maybe 7, 8 years ago, and it's essentially, think of a donut, but a mini donut, right? It's about 10 millimeters in diameter, and it's back to my roots, right? It's a biomaterial sponge-like structure made of silk proteins where we put all of our brain-related cells. So these are human cells go in there, and those cells grow, and they send out their axons into the donut hole, and the donut hole is filled with a gel. So you can sort of see the connections, the connectome, if you will. And I bring it up because early on, a few years ago, we decided to take the brain donut, if you will, and grow patient-derived cells, like from Alzheimer's patients. And you could take these cells, you can reprogram them, as many of your audience knows, and make them sort of grow indefinitely, if you will, in culture. So if we seed those cells from Alzheimer's patients in our donut model, about a year later, that's how long it took, you could start to see phenotypic outcomes that look like Alzheimer's. For example, the things you hear about, plaques would form. But more importantly, you could see the inflammation associated with it. You could measure changes in electrical signals and so on. So we have many, many different markers that would suggest in our donut model in the lab, we would start to see indicators of Alzheimer's disease, which was pretty great, right? And we carried those cultures for more than 2 years, and that's all published. So the good part of that is it worked. You could create a model of the human brain, tissue model, use it in the lab, and you could create a disease state. And that's really important because, as your audience probably knows, for Alzheimer's, Parkinson's, dementia, schizophrenia, traumatic brain injury, depression, you can make the list as long as you want. There are 0 curable treatments today. No one has a clue how to cure any of these diseases. So our mission is to make sure we have tissue models of the human brain that people can use to try and make a difference in those treatments. So the students were great, but a year of culture is painstaking for any student as well as for a PI. And so fortunately, I have a very, very clever postdoc. Her name is Dana. And Dana looked at some early literature from others, including a woman in the UK named Ruth Izaki. And Ruth reported that there was evidence in cadaver brains of viral infections associated with Alzheimer's brains. So what Dana did is she then went back to our donut model and she pre-infected the model with a herpes virus. And if your audience isn't aware, virtually the entire population on the planet is infected with this virus or a version of it. Some of us have symptoms for it, others have no symptoms, but everyone's a carrier. And there's a long history of viral sort of inclusions in our genome. It's nothing new. It's been historically the case. You could argue it's part of evolution, but they're always there. But one of the things that was notable is that when Dana infected our brain models with herpes and then looked at Alzheimer's phenotype development, instead of taking a year, it took literally 5 to 7 days.
Oh, wow.
That means the virus sort of catalyzed or accelerated the development of these symptoms in a dish still. So this is pretty remarkable because now it means we have tools that you could quickly use to screen treatments, whether it's drug discovery or other treatments. This is really, really meaningful on 2 fronts. One, we use the model to start to screen, and we've published this. And there are things like extracts from red wine called resveratrol, things like extracts from green tea. So these are compounds people proclaim may be useful to help with dementia and so on, and Alzheimer's. And in fact, in our model, they actually do really, really well at slowing down the rate of Alzheimer's symptoms that happened in our model. And of the 20-odd compounds we screened, those were the 2 most positive outcomes. Many compounds had no effect. They had no positive effect on slowing down the symptoms. So the good part is the model can now be used to screen, I'll call them nutraceuticals and other compounds, to see if they have value added. And then consumers can decide if this is something useful. This is not a in vivo study. It's just an in vitro screen, but yet it gives you some differences in outcomes that you can start to extrapolate from. And the other part is to think about this in the context of today's world. And that is, we already know COVID has many different symptoms, some of which are brain-related malfunctions. Exactly what those are and what they mean, we don't know. But one of the things we would like to do moving forward is utilize our brain donut model to study COVID infections and then look at what are the downstream effects. Is this the next wave of a new kind of neurodegenerative disease, or is this not something of concern? We don't have the answers. These are things we need to begin to do in our research going forward. So we're really excited about the brain tissue model because of what it offers. It's a very, what we call sustainable model. You could grow it for days, weeks, months, and years, and it still functions beautifully. We could use it to create diseases like Alzheimer's, like Parkinson's, like TBI, traumatic brain injury, all things we're working on. and then start to develop or understand drug interventions in the process. So we're just at the beginning of all that. We see a lot of great future potential.
Well, David, these projects are absolutely amazing. Congratulations to you and your team on just the progress that you've made over the years. And you did touch on some of the challenges with this brain model project. And I think you don't hear about these types of challenges and struggles enough, that it took 1 to 2 years of just waiting and watching. And I think these challenges, these struggles in science need to be shared more so people don't feel so alone when they're having them themselves. So, David, do you have another story of a failure or a challenge you experienced? And if so, could you tell us how you worked through it?
There's challenges every step of the way for any project we have. I could talk about a story, for example, of our genetic engineering of silks as one good example. So one of the things we've done over the years is try to understand mechanistically how silks are prepared, processed, and formed into fibers by spiders and silkworms, right? So you see these beautiful orb webs that I mentioned before, or you see these beautiful cocoons from silkworms that end up in your textiles and things you wear or sleep on and so on. So that's always been something we've worked on for many, many years. We've published on mechanisms that we've learned about and so on. And one of the interesting things is we spend a lot of time exploiting what I call the modular nature of these proteins. So fibrous proteins like silks, keratins, collagens, and the like are very, very what we call repetitive. You could look at one region of the protein and it looks like another region. So you see repeats upon repeats of the amino acids to make up those proteins. That's important because genetically we can then build a module and then just make that larger if we want to encode different protein versions to study these. So naively, we took genes or built genes from the silkworm and built genes from the spider and cloned those into E. coli as our host and expressed the proteins. And we've been doing this for 30 years, just to give you some context. And we have no problem producing spider silks doing it this way. We can produce lots of different silks, reasonable yields, never great yields, but reasonable yields to study recombinant spider silks, to study structure function, build new materials, and so on. Even today, we cannot express silkworm silks very well. There's many reasons we speculate why. It's still frustrating, but it could be that it just precipitates in the cell because it's so hydrophobic. It could be because we don't have all the key elements there, and it could be there might be some toxicity to the cell. My point is science is about things that don't work as well as being observational about things that do work. What we've learned to do is focus on the clones that work well and really build on those as sort of our core domains to build silks of all kinds of variants. We still can't solve the silkworm cloning problem to a high degree, but in many respects now we don't need to because we have sufficiently cloned and expressed other silks to study the properties that we want. So I'm not sure that's a really happy ending, but it's a suitable ending that allows us to continue to move the work forward in new ways nonetheless.
Absolutely. And I think important to just sort of accept that sometimes you're not going to be able to solve a particular problem and you do need to move on. That is definitely an important lesson. And we don't just want to talk about the challenges and the struggles. David, do you have a particular success story that was your all-time favorite, or maybe even just a small win recently that you'd like to share?
Oh, sure. Many of those. Well, I'll tell you a very recent success. So we all talk about the challenge. Well, people call it in different ways, but some people call it the valley of death. So we do some really interesting research, or at least we think it's really interesting research. We think you could actually do something with it to make a meaningful contribution to society, whether it be a medical device, food of the future, tissue model that we talked about. It doesn't matter, right? And so usually you think about these things and it can take decades to get something to that level. But I like to talk about this experience back to our cellular agriculture work. So about a year and a half ago, 2 years ago, we were interested in isolating cells from fish. We had already isolated cells from land animals, a few other things, and we decided we wanted some deep sea fish to isolate cells to then grow those cells up to make, again, foods of the future through cellular agriculture. So the group chartered a boat, a deep sea fishing vessel out of Gloucester, Massachusetts, with a captain on the ship to take us out to the Outer Banks where there's lots of fish here off Massachusetts. And 6 of us from the group went, including myself. And I will say 2 didn't get seasick. 2, like myself, were kind of okay, but on the edge of getting sick. And 2 got very sick. Because you're on very bumpy seas. We're out there for 15, 16 hours. You leave port at like 2 a.m., get back late afternoon. But the good news out of all that, first of all, a great team experience. We still talk about the trip all the time because it was so fun. Number 2, we caught a mackerel and we took muscle biopsies of the mackerel. And those cells turned out to be remarkably resilient because you could spend years, decades, and never succeed at what we call spontaneously immortalizing cells, such as muscle satellite stem cells or fat stem cells towards growing food. Yet in this case, the student Michael was successful in culturing the muscle satellite stem cells from the mackerel and was able to get them to spontaneously immortalize after about 50 doublings. And now those cells are made available through cell data banks to the world to use as a key cell line available to study fish and food production from fish muscle cells that didn't exist before. And that took all of 2 years, literally from start to finish, from going out on the boat to the point we had the cells immortalized till we could sort of license those cells out to a startup company, but also provide the cells to other researchers that want to use them. So I would call that a really fun, positive success because there's other cells we've tried to isolate for years now without being able to spontaneously immortalize them. In some cases, not even able to isolate them. So you never know, right?
Oh, well, that is such an exciting success. Congratulations to you and the team there. And this sea adventure sounds phenomenal. And I know it's easy for your science to sort of take over your life. You're spending so much time there in the lab because you're excited about the work. But we always encourage our readers to maybe take a step back, to try to read broadly, to take a break and expand their minds. So we always love to get book recommendations from everybody we have on the show. So David, do you have a favorite book that you can recommend for me and our listeners today?
So I'm an avid reader of books about trees, going back to my roots, right? Pardon the pun. So there's a couple of books, Wild Trees, Secrets of Trees. You could go on Amazon or wherever your favorite shopping spot is. I would encourage you just to read about trees in any of these books because you can approach trees from a very scientific perspective just to learn about structure function. But most of these books are more either historical. So there's some of these books, Secrets of Trees, go into cultural relevance of trees and how they've played a role in religion and culture for eons. And I find that fascinating. And the other side of it is learning about how people interact with trees, what people do around trees to think about how meaningful they are in our lives as well. Those are the books I would recommend. Anything you can find and read about trees as something that would really, I shouldn't say that, but I would hope it would broaden your perspective about how significant and important trees are in everyday life in ways you may not think about.
I love it. Well, David, we will add some of these recommendations to our website for our listeners to find there. And we also talked at the beginning of our show about how you love to travel. And I think this is a wonderful aspect of careers in science. You get to go to conferences, work with collaborators, and see the world. So do you have a favorite place that your science has taken you?
Maybe the most intriguing, enriching place for science was in Mauritius, which is a small island in the Indian Ocean off Madagascar, Africa. And I went there maybe 15 years ago, plus or minus. We were fortunate to acquire an NSF, National Science Foundation grant to teach the field of biomedical engineering at the University of Mauritius through a colleague I had there who was a polymer scientist. And I didn't know much about the island, but certainly was intrigued to go. And I asked 6 of my colleagues to join me. So it was a team to go there and teach the field, which was great. But as we were on the island, we learned science from all perspectives and in ways I never expected. So first of all, we were on the edge of the island at a resort. So you were sort of in an artificial paradise, plentiful food, et cetera. It was really interesting and very comfortable. That was great. And then we would take tours of the island, and they had a zoo on the island. And from a science perspective, the zoo was remarkable because at the zoo, first of all, there was a display of insects that they had captured on the island. And in my life, I have never seen creatures like this, right? They just, number one, the size of these insects told you how they had evolved on this island away from a lot of other pressures. The size was remarkable. The colorations were remarkable. Species I had never seen before. So that alone was eye-dropping to me, just to see the diversity of insects that had evolved on this isolated island. And the other was, it was my first time in my life I had been able to literally be with the giant tortoises. I had read about them and seen them in videos, but to be able to literally touch a giant tortoise, watch its motion, to think this apparently clumsy creature yet lives this long, has lived away from all these environmental whatever's going on out there, is just remarkable as well. So those are things scientifically, I think, that really opened my eyes to, again, the further diversity of creatures on the planet, and how when they're away and isolated, you could see these remarkable creatures evolve in ways you would never expect. I think that was eye-opening to me from a science perspective. The last part of this island, which I never knew about till I was there, was one of the reasons we were invited to the university was that they had built a brand new hospital in the very center of the island. And we were asking questions about this hospital, and it turned out this is what I would call medical tourism, health tourism. There are other terms for this, but this is an isolated island where private investors built a shiny new island for people to fly in from all over the world to get stem cell therapies. And what was eye-opening to me about this from the science, and I'll call it the darker side, is that there's no documentation of anything they were doing there that provided any benefit to any patient, because we asked.
Oh, wow.
Yet people were willing to spend a fortune to fly from anywhere to come to the island, stay at a luxury hotel, but get stem cell treatments at this hospital for a very high price, right? And so I bring that up because as scientists, I think we have an obligation to educate consumers about what's real or not real. And science today, you just wonder, it gets under attack, not under attack. And so I think it's a good case of sort of privatization of this kind of scientific treatment without really any scientific basis for it. Yet people will come if they're desperate. And we need to all be aware of that and do our best to try and make sure consumers are aware of what is real or not real. So in this one island, I learned so much, right? I learned about diversity and amazing creatures. And I also learned about a darker side of science that I hope we can continue to work on and improve those outcomes going forward.
Absolutely. Well, you brought up so many great points in this response, David. I think those ethical considerations are something important for us to continue working on in science and as a society as a whole. And I think oftentimes though, science and scientists are portrayed as being very serious, very critical. There's this sort of stereotype about life as a scientist. And I know research is very important, but some of the funniest and most creative people I've ever met are scientists. And I think everyone's got these sort of quirks and maybe strange traditions in the labs that they run. So David, do you have one of these traditions yourself, or maybe a story that really shows this human side of science that people don't often get to see?
I guess I'd answer it a different way, probably not what you expect, because I'm certainly quirky. I embrace my quirkiness, and this is the age of the nerd, so we're all happy that nerddom is acceptable these days. But I always say to my students and others that you've got to have a sense of humor in life, because if you don't have a sense of humor, life can be probably long and difficult. So I always think comedy or those kinds of reliefs are really, really important. And I can tell you in my own research group right now, we have a couple of stand-up comedians who go out and do open mics and things like that. So I think your point is very well taken. I think a sense of humor can balance a lot of other things in life. And if you learn not to take yourself too seriously and embrace these differences, I think you'll enjoy life a lot more. And I think it just gives you a path to be open to these changes, the zigzag we talked about, and open to learning more than you would be otherwise. So I would just amplify on what you said, that I think humor, sense of humor really can go a long way to helping you enjoy your life more and more.
Definitely. So does the group get together to attend these open mic nights?
Some, yes. And certainly at our holiday parties, there's plenty of fun to be had. Let's put it that way.
That is phenomenal. And I think you are absolutely right. You need that sense of humor. You need to not take yourself too seriously because the work is so serious. You're answering some of these really difficult, large problems in your laboratory. And oftentimes things can hold you back, whether it's funding, staff, technology, feasibility, or other challenges that you run into. So if we took away all the barriers that normally hold you back, David, what is the one question you would want to answer or problem you would want to solve?
This may come out of left field. Because of the work I talked about the human brain, I had, again, another very creative postdoc who was interested in learning. So Nick, who's now up in Canada, basically took our brain tissue models and demonstrated, this is published as well, that you could sort of electrically stimulate the brain model in different ways. And based on the changes in response, so think of excitation response from the neural networks in the brain model, you could argue based on different definitions that the brain model was quote unquote learning. So one of the questions I'm intrigued by is, could you in fact build brain tissues like this and teach them things? And then let's say you have a patient with dementia, or you have a patient that had brain surgery, or maybe you just want the future people to have an opportunity to switch out their brain modules to be really good at something for a while and then do something else. Could you eventually develop brain tissue with learning modules in them? So the future computer, if you will. Can you do this? I have no idea. Am I intrigued by it? Yes, because I find the brain is really this last bastion of complete unknown about the details of how it works, how memory really works, how learning really works, and how can we take that understanding and turn it into something to help mankind, humankind. And so if you ask me that question, that's probably my answer, that I would love to see how to develop that level of understanding to really make a difference in all the brain-related functions that we deal with today, whether it's the brain diseases again, or brain aging in general, or learning in general to help people.
I love that. And we appreciate you giving us some big ideas to ponder today. And you've shared a lot with us today, David. I'd love to end with advice. So is there a piece of advice that really helped you that you can share with me and our listeners today?
It all boils down to passion, whatever you're doing in life. And so I always say, read stuff until your heartbeat goes up, and then that's what you focus on. Find something you're passionate about, something when you're reading, you stop and really dig in more detail about, and find a way into that field, whatever it is. And even if you know nothing about it, it doesn't matter. Find a way to get involved and get help from those who can mentor you, use networks, whatever it is. But if you find something you're passionate about, it never feels like a job, right? And it's something you'll do 24/7 and would never feel like you're working. So I guess that's my advice, is try and do that. I feel like a very fortunate individual that I was able to find my path. I know people out there who never find a path. The sooner you can find something like that, the better you'll be, the happier you'll be, the more impact hopefully you'll have on other people and society. So go with your heart is what I would say.
Well, wonderful advice. And we appreciate you sharing that with us today. Is there any last message or last note of inspiration you'd like to leave us all with at the end of our conversation today?
No, I think I said a lot, but I'm always available if someone wants to connect and talk further or get some advice or re-educate me on something I missed or failed with here. Please don't hesitate to reach out. I'd love to have a chat.
Wonderful. If our listeners want to connect or want to learn more about you and what you do, David, what's the best way for them to do so?
Probably the easiest way is just go to the Tufts University website and put in my name, Biomedical Engineering. My website will pop up, and then you can connect with me from there or read more about what we're doing, and we can go from there.
Wonderful. Well, listeners, definitely check out that website. David, thank you so much for joining us today. It's been such a pleasure to chat with you.
Thanks very much, Marie. It's been my pleasure to be on with you today.
Wonderful. And listeners, great to have you here with us as well as always. We hope you'll join us next time for another episode of People Behind the Science.